program for postgraduates in pathology. Today's talk will be a slide seminar on flow cytometry and molecular pathology. The presenter are Dr. Mayur Parihar, who is a senior consultant hematology. Will be an interactive session, and the postgraduates who are there in this session are Dr. Rana, Dr. Sushma, Dr. Nishta.
Good evening, everyone.
These are the youth May, and on 25th May, there is a slide seminar on flow cytometry and molecular pathology by Dr. Shweta Rajpal and Dr. Gaurav Chatterjee. Let's start today's session. I welcome Dr. Mayur Parihar and Dr. Ashish Rath.
Good evening, everybody. So till Ashish shares his slides. So Ashish and I, we've got together to bring in some interesting cases. And I think Ashish is going to start presenting by sharing the slides. Yeah, Ashish, over to you.
Yeah. Is it visible now?
Yes, sir.
Yeah. Okay. So, good evening, everyone. So, let's start our session for today. So, let's start with the case one. So, case one is of a 62 years male. He had weakness since 1.5 years and non-healing ulcers over lower extremities and left ear pinna. He was evaluated extensively outside with multiple hemograms, CT scan, echoes, colonoscopies, and bronchoscopies. However, no positive findings were ever found. He had a history of COVID-19 infection in November 2020. However, that was a mild symptomatic COVID. There was no previous history of any malignancies, no significant family history. And because of these non-healing ulcers and presence of mild anemia in the patient landed up in Tata Medical Center, Kolkata. So at TMC, we found that these type of non-healing ulcers were there in the lower extremities as well as on ear pinna, as you can see in these pictures. There was absolutely no lymphadenopathy, hepatosplenomegaly. However, there was mild anemia in the patient. So we went on to do a CBC, which we do when the patient land up in the hospital, and you can see the hemoglobin was just 9.9. So it was not very severe degree of anemia. Mild pallor was there in the patient, and which corroborated with this hemoglobin. However, total leukocyte count was almost around 30,000 with normal platelet count and almost normal retic count. There was a neutrophil predominance. And this is the type of peripheral smear which you can see where there are hardly any WBCs there. As you can see, the TLC is 30,000. I would have expected on a 48 there would have been some 3 to 4 to 10 WBCs in the peripheral smear. However, we can hardly see any peripheral smear increase in WBCs. So is it really leukocytosis? So how to define this leukocytosis? Yes, this has to be more than 11,000, but on peripheral smear, that is not 11,000. So peripheral smear manual counting come out to be just 8,000. So possibly this is a pseudoleukocytosis. However, when we check CBCs and peripheral smears more on peripheral smear, we could find these bluish colored deposits. So Dr. Rana, can you tell me what kind of deposits these are?
They can be cryoglobulinemia.
Okay.
Or cold antibody deposits.
Okay. So before even jumping into the cryoglobulins or cold antibodies, first you have to rule out artifacts whenever you see this kind of deposits in a peripheral smear, which are more common than that of presence of cryoglobulins. So what kind of artifacts can take this kind of staining? This can be just stain deposits. Whenever you do a staining such as Leishman stain or a Leishman James stain, your hematoxylin will stain or deposit like this, bluish color deposits. Or there can be sometimes presence of fibrin whenever the clot activation is there. The fibrin can also take up the stain and can be stained like this. So you have to rule out the artifact. So to rule out artifact, you can again collect the sample, a fresh venipuncture sample, again prepare a peripheral smear and stain it. So we did that. So a repeat sample was ordered and staining was repeated, and again the same kind of deposits were there in the peripheral smear. So now there is a suspicion that this can be a cryoglobulin or cold induced. So what we did, we incubated the sample to find out if it is really cryoglobulins. So what happened? So previously, the amount of these deposits were more. However, after incubation, the amount of these deposits decreased. So we knew that this deposit is due to presence of something related to cold. However, there was no RBC arbutination. To our surprise, when we incubated the sample, the TLC came out to be 9,500. However, previously it was 50,000. So the pseudoleukocytosis was confirmed. So what can be the diagnosis, Dr. Anna?
Cryoglobulinemia.
Yeah, this can be cryoglobulin, but before jumping into again the diagnosis of cryoglobulin, you have to prove that this is cryoglobulin. So you have to do a cryoglobulin test. So to
Yes.
do the test, what you do, you take two vials, that is red of vials, where there is no anticoagulant. You take red of vials and transport it to the lab at 37 degrees Celsius. And always remember that transportation has to be in 37 degrees Celsius. That is the body temperature. Once it reaches the lab, you keep it for 1 hour in 37 degrees Celsius in the water bath. Let the clot form. After the clot is formed, you centrifuge the rectal vial, take out the serum, and you put this serum into a glass tube as well as a Winthrop tube, and keep it in 2 to 4 degrees Celsius in the refrigerator. So once this turbidity forms, this cloudy turbidity forms, you know that this kind of cryoglobulin is present. However, simultaneously, you have to do the test on a patient plasma also, because there is something called cryofibrinogen which can give rise to these kind of symptoms. So to rule out cryofibrinogen, you have to do the test both on patient plasma and patient serum. If it is present in both serum and plasma, it is cryoglobulin. If it is present only in plasma, not in the serum, it is cryofibrinogen. Simultaneously, you have to put a control serum also to find out if your control serum also has same kind of deposits or do not have same kind of deposits. So once you have confirmed that this patient serum has a deposit which is cold-induced or cryoglobulin, you have to measure what amount of cryoglobulin is there in your patient. So you take a Winthrop tube, fill it up to the mark, and keep it in the refrigerator. Then once the cryoglobulin is formed, you centrifuge the Winthrop tube. The amount of deposits will define your cryoglobulin amount. That is called the cryoglobulin. So once we have confirmed that this is cryoglobulin, the diagnosis is now cryoglobulinic vasculitis.
Okay.
So there are different types of cryoglobulins. So what kind of cryoglobulins are there? There are three types of cryoglobulin according to this classification. This can be type 1, type 2, or type 3. So type 1s are monoclonal immunoglobulins. Type 2 are monoclonal mixed with polyclonal immunoglobulins. And type 3 is purely polyclonal immunoglobulins. So type 1 mostly occur in malignancies such as Waldenström macroglobulinemia or multiple myeloma or any lymphoproliferative disorder or light chain diseases. Type 2, where there is a monoclonal immunoglobulins mixed with polyclonal immunoglobulin, it can be due to hepatitis C, which is most common cause of cryoglobulins, and there can be sometimes autoimmune disorders like Sjogren's syndrome or rheumatoid arthritis. Polyclonal cryoglobulins are mostly due to autoimmune disorders or some infections. So now that we have cryoglobulin in this patient, a cryoglobulin vasculitis is there, we have to know that what kind of vasculitis it is. So type 1 monoclonal immunoglobulin causes vasculitis, which is mostly due to your vessel obstruction rather than a complement activation. However, your mixed cryoglobulin, which is most commonly due to hepatitis C virus infection, you can see a leukocytoclastic type of vasculitis, which is due to immune complex deposit and complement activation followed by inflammation in the endothelial cells. So you can clearly see whenever you get a leukocytoclastic vasculitis, the typical appearance in case of your cryoglobulin vasculitis is like the picture stored here. However, whenever there is a vessel obstruction related vasculitis, you will see that small vessels will get obstructed. You may find acrocyanosis or this kind of ulcer in the ear pinna. So there, we did some additional tests like hepatitis C, which was negative. Respiratory viral pathogen also were done to find out if it is due to some virus infections, which were again negative. The ALA was negative, CP4 was normal, so we ruled out the autoimmune diseases also. However, we found that the IgM, serum IgM is elevated. It was around 950 milligram per deciliter. And on serum protein electrophoresis, we found 2 PET M spike in the gamma region. And immunofluorescence electrophoresis also showed these proteins to be IgM kappa, or confirmed that these are monoclonal. So now, once we have found that this IgM is increased and this is IgM kappa, or this is a monoclonal protein, and the patient is symptomatic. So now, what next steps should be done, Dr. Rani?
Bone marrow exam and immunohistochemistry.
Okay, so we went on to do a bone marrow examination. Can you say what are the findings in the bone marrow?
First, we see a hypercellular marrow. We see many lymphoplasmacytic cells and occasional plasma cells.
So as Dr. Rana said, as you can see, there are a lot of lymphocytes which are increased, and along with lymphocytes, there are plasma cells which are there in the bone marrow. However, apart from lymphocytes and plasma cells, there are such cells where we cannot distinguish exactly between a lymphocyte and plasma cells. The nucleus is eccentric. There is some kind of hop is there, but this doesn't exactly look like a plasma cell. So this kind of cells, which is in between a lymphocytes and plasma cell, these cells are called lymphoplasmacytic cells. So in this marrow, we found small lymphoid cells. plasma cells as well as lymphoplasmacytic cells. So the most closest diagnosis will be
Plasmacytic lymphoma, LPL.
so lymphoplasmacytic lymphoma. Now that we are suspecting lymphoplasmacytic lymphoma, we have to again confirm on bone marrow biopsy that this is a lymphoma. So before jumping into bone marrow biopsy, in this picture you can see some kind of cells are there. So I'll request Dr. Susma, can you tell me what kind of cells these are?
Mast cells.
Yeah, these are mast cells. How do you know these are mast cells? Because you cannot differentiate between the cytoplasm and the nucleus. And you can see a pinkish hue around these cells. Whenever you find this kind of cells in a marrow aspirate, and you find that there is a pinkish hue around these cells, you confirm that these are mast cells. So there are mast cells, there are lymphoplasmacytic cells, increase in lymphocytes, plasma cells, and these kind of clusters of plasma cells. The diagnosis will be done on a bone marrow biopsy now. So, Dr. Ast, can you describe what are the findings in the bone marrow biopsy?
Yes, sir. So the biopsy shows paratrabecular nodular aggregates of monomorphous population of lymphoplasmacytic cells. The cells are quite mature with condensed chromatin. The IHC highlights CD20, which is a marker for mature B cells. So this favors a diagnosis of mature B cell neoplasm. And CD3 does not highlight the lesional cells. Just the scattered T lymphocytes are highlighted by CD3.
Thank you, Dr. Asta. So as Dr. Asta has already said, you can see there are these nodular aggregates of lymphoid cells which are there in the bone marrow biopsy. Again, you can see there are these small lymphoid cells. How to know that these are small lymphoid cells? As you can see, the nucleus are very close. That means the cytoplasm is very scant. Whenever the cytoplasm is scant, the nucleus will lie close to each other. So these are small lymphoid cells. However, there are cells which are eccentric nucleus and there is a lot of cytoplasm. So there are some plasma cells, some lymphoplasmacytic cells, as well as small lymphoid cells. And these cells are highlighted by CD20 and CD3 is negative in these cells. But what about the plasma cells? As we know that lymphoplasmacytic lymphoma is the malignancy of small lymphocytes, plasma cells, as well as lymphoplasmacytic cells. So your lymphocytes as well as your plasma cells have to be clonal. Now that you have proven that your B cells are increased and they are CD20 positive, you have to prove that your plasma cells are also clonal. And if they are not clonal, probably this is not LPL or Waldenstrom's. So on CD138, you can see there are some aggregates of plasma cells. Again, you can see some aggregate of plasma cells, and these plasma cells are taken up kappa. So as you can remember that on our immunofixation electrophoresis, the clonality was kappa. It was IgM kappa. So again, our plasma cells showed a kappa restriction. Lambda was scatteredly positive in these plasma cells. So we know that there is an increase in CD20 positive cells. CD130 also show aggregates which are kappa restricted. So I know that my bone marrow biopsy has increase in B cells which are in nodular aggregates. There are some lymphoplasmacytic cells and my plasma cells are also clonal. So I certainly know that I'm dealing with a LPL or a Waldenstrom's macroglobulinemia. Whenever in the bone marrow aspirate or in the bone marrow biopsy, you see increase in mast cells, you are more sure that you are dealing with LPL because mast cell prominence is commoner in LPL rather than in other low-grade malignancies. So we went on to do a lymphoproliferative panel by flow cytometry. As you can see, there are 4 different panels. So one of the most important tube is the tube 3, where there is a lambda and kappa, which will define your clonality in the B cells, and there are other cells to distinguish between different low-grade and high-grade malignancies. So we went on to do a flow. And Dr. Susma, can you describe what are the findings on the flow cytometry?
Actually, in the flow cytometry, so the positive findings are CD45 is in moderate and kappa is positive and CD23 is positive. CD103 is negative, CD200 is negative, CD5 positive, and CD123 is negative, CD11 is positive.
Okay. So as you can see, the first plot or dot plot which we do in the flow cytometer experiment is a 45 band side spectrum. As you can see, there is a increase in cells in the 45 bright region. So usually in the 45 bright region, the lymphocytes lie. And these are the cells marked in red are granulocytes which have a moderate 45 and high side spectrum. So once you have detected your lymphocytes, I know that lymphocytes are increased in the bone marrow aspect. And I know that on my bone marrow biopsy, there is a CD20 positive aggregate. So I get the CD19 positive cells because I'm suspecting of a B cell malignancy. So once you get to CD19 cells, look at your CD20. So CD20 is very bright in these cells. And then define your clonality on a kappa and lambda, which is surface tense for light shades. You can see there is a kappa restriction. All of my B cells are positive for kappa. So there is a kappa restriction. You can see the 5 is negative in the B cells, which are highlighted by blue. CD23 is very dim positive, only in a subset of the cells. Again, FMC7 is almost negative, CD10 is negative, CD200, as you can see, almost all cells are dim positive. 25 is again positive, and 11C and 123, which are the markers, 103, which are the markers for hairy cell, are negative. Again, 79B, very bright. You can see there is some amount of CD38 which is positive. So these CD38 positive are nothing but the lymphoplasmacytic cells and the plasma cells which we saw on the bone marrow aspirate. As you can see, again, CD43 is very dim and IgM is bright. So in my flow cytometry experiment, now I know that my B cells are kappa restricted. They're expressing very bright 20. They're expressing 38. They're expressing surface IgM antibody, and they're kappa restricted. However, the 23 is almost negative. 25 is positive. So I know my B cells are clonal, but they are not CLL. They are not marginal zone lymphoma because CLL will have an expression of 5 and 23, and my marginal zone lymphomas usually they will express 23, and usually they will be negative for 25. But again, before jumping to diagnosis into LPL, diagnosis of LPL, I have to prove my plasma cells are clonal. So we went on to do a plasma cell tube where you can see you get your plasma cells based on CD138 and CD38 positivity. You saw that your plasma cells are 19 and 45 positive, and they are 56 negative. However, again, these plasma cells are positive for kappa, that is kappa restricted. So what is my final diagnosis? Yes, my final diagnosis will be lymphoplasmacytic lymphoma. As the serum IgM is increased and there is IgM kappa, my diagnosis will be Waldenstrom's macroglobulinemia. So there are certain flow cytometric features which are characteristic of LPL or a Waldenstrom's. It is usually 5 and 10 negative. You will find a brighter 9, brighter 20, 79B will be bright, IgM will be bright. Usually 25 will be positive, 38 will be positive. However, very few cases will express 23, unlike your CLL or marginal zone lymphomas. And these CD200 is frequent in this LPL and Waldenstrom's. However, you will not find a bright 200 unlike the CLL. Again, the plasma cells, unlike myelomas, where you define your abnormal plasma cells to be 19 and 45 negative, they will be 56 positive. In Waldenstrom's, your plasma cells will almost always be 19 and 45 positive, 56 negative, but they will be clonal. So just because of 19, 45 positive, this will not be normal plasma cells. You have to define your clonality in your plasma cells, or you have to demonstrate your clonality in these plasma cells. So after I have known that this is LPL, again, to prove it to be LPL, there is something else which we need to do. So Dr. Askar, can you say what next should do in case of LPL?
So the mutation study that is made 88 needs to be confirmed to confirm the diagnosis.
Okay. So we went on to do a molecular. I'll request Mayur sir to take over now to let you know the part of molecular.
Yeah. So I think so by now Ashish has more or less convinced, I mean, not more or less, it's completely convinced everybody that it is an LPL. But sometimes, in this small round cell lymphomas, you need molecular sometimes to confirm your diagnosis. And what we have done here is an MYD88. So what is MYD? It's a myeloid differentiation factor. Now, this gene actually codes for protein, which deals with signaling in immune cells, and that is why it is seen in lymphocytes. So what does your MYD88 do? It acts as an adapter protein and actually passes the signals that come from the surface of the cell downstream. So what we have done here is we do what is called as an allele-specific PCR. So it is a very specific PCR wherein we use two different sets of primers, one for the wild type and another for the specific mutation, which is L265P, which is at position 265, leucine has been replaced by proline. So Ashish, if you can point to the patient mutant there with your pointer. Yeah. So what you see here is basically you have a wild type and a mutant. So basically it's a heterozygous mutation. So you'll have one normal copy of the gene and one gene will be mutated. And we have added two different sets of primers so that to see whether what part actually amplifies. So what you see here is when we ran it in the gel, you see an amplified product both for the mutant as well as the wild type, clearly showing that it's a heterozygous MYD88 mutation. And of course, when you run a PCR like that, you have to take in the positives and the negative controls have to be put in the run that you do. Next slide. So as I said, what does MYD88 do? It is an adapter protein that helps in passing the signals that come from the membrane to the downstream. It is seen in 90 to 95% of LPLs, and it is not a completely diagnostic of LPL. Of course, you have to correlate with your immunophenotypic features or immunohistochemistry because these could be seen in splenic marginal zone lymphomas of CLL in a minority of about 3 to 5%. 60 to 80% of them have IgM, and 30% of cases of ABC DLBCL can also show this MYD88 mutation. Now, so this is helping in diagnostic. Do you do any other molecular study? Yes, you can do a CXCR4. The patients with about 30% of LPLs have a CXCR4 mutations, and patients who have these mutations usually have resistance to BTK inhibition by ibrutinib, and these patients have a poorer prognosis compared to those that do not have the CXCR4. Next. So as I said, it is not completely diagnostic. It could be seen in other MYD mutations could be seen in 5% of SMZL, ZZL, or nodal marginal zone lymphomas could be seen in 90% of LPL. And the other genes, when you look at the mutations, you have BRAF mutations, which are seen in hairy cell leukemias, and the MAP kinase mutations in the variant hairy cell leukemias. Next slide, Ashish. Next.
Yeah.
So before we go to case 2, I think it is open for any questions regarding this case from anybody. If you have any doubts. Are there any questions on the comment section, on the chat section? So Ashish, there is a question from Jayashri which says, what is normal kappa lambda ratio in a man?
Okay. So, thank you for the question. And when you talk about the kappa lambda ratio, it varies based on your ISCs or flow cytometry. So when you talk about ISCs, the safest kappa lambda ratio which will diagnose the clonality will be 5:1. Anything which is more than 4:1 for kappa lambda or 1:4 for a kappa 2 lambda will be diagnostic of clonality. However, it is safer to go for 5:1. However, in a flow cytometry experiment, there is always 4:1 or 1:4 for diagnosis of clonality in case of kappa and lambda. But you will hardly find any cases where there will be confusion in case of a CSO lymphoma or a myeloma. There will be hardly any light chain clonality which will be confusing because you will always find clonality to be either kappa or lambda. However, yes, there can be some cases where both will be there because of intermingled normal plasma cells or normal B cells. However, it is always safe to go for 4:1.
Any other questions? If not, we'll move to the second case. And if anybody has any questions regarding the first case, they can ask us even later. Ashish.
Yeah. So we move on to case 2. This is a 45 years male who presented with cough and disease in 3 weeks. Outside series showed pancytopenia. However, he was referred to TLC with a suspicion of myelodysplastic syndrome. At Tata Medical Center, we found that again only pallor was positive, no organomegaly, no lymphadenopathy. And on peripheral smear, as you can see, there is anemia, there is a leukopenia, and the platelet count is again dropped, which is 28,000. And you find these kind of cells. Dr. Sneha, can you tell me what kind of cells these are?
These are leukemic promyelocytes containing rods.
Okay. So whenever there are multiple auras, what do you call it?
Faggot cells.
Yeah, faggot cells. So as you can see on peripheral smear, there were multiple blastoid cells or blast-like cells. So you can see the orders, and in some of the blast, you can see multiple orders, which are thick and stout orders. Whenever you describe orders in classical cases of translocation 821 leukemias, you will find thin and slender orders. However, in classical promyelocytic leukemias, you can find these thick and stout orders. So this is how we describe the orders in case of acute promyelocytic leukemias. Then again, we went on to do a bone marrow. Again, in bone marrow, you can see there are a lot of these abnormal promyelocytes, which have eccentric nucleus in very granular cytoplasm. As you can see, very granular cytoplasm, and there are multiple faggot cells which are present. Even on bone marrow biopsy, you can see one of the bone marrow biopsy, the abnormal promyelocyte is showing some of the all rods. So sometimes, yeah, you can see faggot cells on the bone marrow biopsies too. So once we have done the bone marrow aspirate and bone marrow biopsy, and we are suspecting a promyelocytic leukemia, so know the different morphologic variants in case of acute promyelocytic leukemia, because it is usually common to find faggot cells where it is easy to diagnose acute promyelocytic leukemia, but there are a number of cases where you may not find a faggot cells or even granules. You may be confused with the diagnosis of promyelocytic leukemia to be made or not. There are different morphologic variants, so broadly it can be hypergranular or hypogranular or a hyperbasophilic one. However, if you go to this paper which was published in 2000, there are multiple morphologic variants in case of leukemia. So in case of promyelocytic leukemia, you may find the faggot cells. Sometimes you can find a pseudo-legacy-like granules in the promyelocytes. Sometimes your promyelocytes may just look like a myelocyte which can be there, and sometimes you may just have a hypogranular variant where you can find a grooving in the cells, which classically we call it as buttock cells or sometimes angle wing cells. So you have to remember that sometimes the classical 11-17 translocation APMLs may show maturation beyond promyelocyte stage. So the classical teaching has been that in case of promyelocytic leukemias, you may not find mature cells beyond promyelocytes, but whenever there is a translocation 11-17, you may show the slide may show maturation beyond promyelocyte stage. But in those cases, the neutrophils are almost always dysplastic and all rods may be absent. So you should be extra vigilant to diagnose promyelocytic leukemias in those cases, because once you raise the suspicion, then the further test will be done to diagnose it and the patient may be saved. So we went on to do a flow cytometry. So this is the acute leukemia panel which we do. We first run a screening tube to find out if it is a myeloid or a B cell based on CD19 or a T cell based on CD7, and then further, as soon as we know that this is myeloid or B or T, we go on to do BLN tubes or myeloid tubes or TLN tubes. So we did flow cytometry in these cases, and you can see first we get the singlets, which are only single cells which we analyze on flow cytometry. Then you get the viable cells, so you get out the low power scatter and side scatter, which may be fragmented RBCs or dead WBCs. Then you do a 45 side scatter as on the flow cytometry experiments, and you find that there is a pink population which is CD45 derived, which are lymphocytes. And again, you have found that there are a lot of promyelocytes, but the typical blast window in case of AML lies in the moderate scatter and dim region. However, here all of the cells were having a very high side scatter because the promyelocytes had too much of granules in their cytoplasm. So the typical appearance in case of scatter, as you can see, this is a teardrop kind of thing. So whenever you typical promyelocyte, you do a flow cytometry, you will find the teardrop kind of scatter in a CD45 and side scatter plot. And you have to define again your autofluorescence level, because these promyelocytes have a lot of granules. So even without antibodies, they will have their inherent autofluorescence. So once you have run your unstained tube where you have not put any antibodies, you know that what amount of fluorescence your unstained tube is showing. So once you know that your unstained tube's promyelocytes are showing certain kind of fluorescence, you have a cutoff for your autofluorescence. So if it is beyond that autofluorescence level, will be called positive. So here my promyelocytes were just 117 positive, were negative for 34. Again, they are positive for 30, which is heterogeneous. Why heterogeneous? As you can see, this is almost going from log 0 to log 2. So there is almost 2 log expression of this promyelocyte. This is the varied expression or heterogeneous expression for 30. As you can see, again, 19 is negative, CD117 is positive, CD7 is negative, which is a T cell marker. MPO is very bright positive. Again, you can see the 33 is very bright positive, very, very bright, much brighter than the granulocyte which are there. How do you know these are granulocyte? Because these red cells show 15 and 33 and are intermingled with the promyelocytes. So once these show 15 brightness, we know that is a granulocyte. So compared to this, these promyelocytes express a bright CD33. They are negative for 15, they are negative for 14 and 64, which are monocytic markers. And again, you can see that there is a 38 positivity and almost negative 11C. As you can see, there is a granulocyte which will be 11B positive and very dim 11C positive. Compared to that, the promyelocytes are negative. So based on that, we know that there are certain typical features of acute promyelocytic leukemia on flow cytometry. So it will be most commonly 34 and DR negative. So Dr. Kusma, can you tell me what are the other leukemias which can be again CD34 and DR negative?
The pattern, can you please repeat the question?
Yeah. Are there any other AMLs or acute myeloid leukemias which can show a loss of HLA-DR and 34? Yeah, there are again other leukemias which can, yeah. So other leukemias which can be there. Yeah. Again.
M4 and M6 form.
Okay, so unlike you're told that monocytic leukemias, but monocytic leukemias will almost always show a brighter HLA-DR because these are monocytes. They are innately HLA-DR positive. But sometimes you can have NPM1 mutations where there can be again 34 and DR loss, which is common. But your NPM1 mutated cells will have a nuclear coupling. So what are nuclear couplings? These are cytoplasmic invaginations into the nucleus. So they will appear like a pale area in the nucleus. These are called nuclear couplings. So whenever there is a nuclear coupling, you don't find any OR rods and your 34 and DR negative. Possibly these are NPM1 mutated AMLs. The classical features in case of acute promyelocytic leukemia in flow cytometry is a DMA 45 with very high scatter, which is called a teardrop pattern or a plane pattern. There will be frequent absence of 34 and DR. Almost all cases will be 34 and DR negative, but remember that there are some cases which will be 34 positive, which are not classical PML RARA fusion females, and there can be certain cases which can again show HLA-DR positive. So absence of 34 and DR though is diagnostic, there can be cases which will be 34 and DR positive, and you should not rule out APMS based on the 34 and DR. CD117 again can be positive. It almost always shows a heterogeneous 13 and a homogeneous bright CD33. APMS are usually 11B, 11C, and 66 negative, and there is frequent absence of CD15, which is a maturity marker which comes after myelocyte stage. So your promyelocyte stage will show a dim 15, so it may show a very low expression of 15. However, it will never show a bright CD15 as such. Your promyelocytic leukemia may have aberrant CD2 or CD56 expression, but remember, if your patient has got ATRA from outside somewhere, it may upregulate 11B, 11C. So take the history if your 11B, 11C is positive again in case of where there is a 34 and DR negativity. If there is a history of patient has taken or translated nucleoside or ATRA, it may upregulate 11B and 11C. You should keep that in mind. And again, as I told, that NPM1 mutated AML or sometimes this KMT2A or MLL enhanced AML may show APL like immunochronic that their HLA-DR might be lost. However, you should remember that once you suspect a case to be promyelocytic leukemia, your flow cytometry is not diagnostic. It may show some characteristic feature, but it is never diagnostic. Once you have known that your promyelocytic leukemia is there, you should go for some molecular test. So, Dr. Rana, can you say what molecular test you should go for whenever you see a promyelocytic leukemia?
Do fish to detect PM-RARA fusion.
Okay, so I'll now request Dr. Ramesh to take over and tell you about how to do a piece of.
Yeah. So as Ashish was saying that sometimes you do have even morphological features and even immunophenotypic features that may mimic an acute promyelocytic leukemia, especially the NPM1 mutations or Kapsche-Blast, and sometimes these cells do show rods, and when you do even a cytochemistry NPO, they are very strongly positive. So in our hospital, every time an acute promyelocytic leukemia is suspected, a fish is ordered, or we move ahead fishing looking for the PML RARA fusions, and usually the report is released within 4 hours. So it's kind of an emergency in our hospital, in our setup, whenever you have a suspected APML. And I always say that the APMLs always come on weekends. So when you're going home on a Saturday evening, an acute promyelocytic leukemia patient will just walk in. So what you see here is, I think Ashish may have to point the fish. So what you see here, the blue color is the nucleus, and it's stained by DAPI. And we have put a dual color, dual fusion probe. So whenever a dual color, dual fusion probe is put, whenever it is positive, you will get yellow color, you will get a fusion. What we saw in our patient was that there was no fusion, but the RARA is showing 3 signals. So you can see that there are 3 green signals here, saying that, okay, the RARA has rearranged, it has gone somewhere, but it has not gone and met the promyelocytic leukemia, the PML gene. So probably there could be a variant RARA translocation. So whenever you suspect a variant RARA translocation, the next step you do is you do a RARA break apart probe. So how do break apart probes work? So the break apart probes are designed in such a way that the red and the green probes are together at the normal RARA gene. And every time there is a rearrangement, the red and blue get separated. So what you see on the right side is an inverted dark image, and you can see that the 3' RARA has actually gone, the red color has gone to a long chromosome. So we are now sure that we are dealing with a variant RARA translocation. So once you kind of confirm that we are dealing with a variant RARA translocation, it is essential to identify the partners. So one of the ways to identify the partners is, of course, metaphase fish, or you can always do the karyotyping. So next slide. So what you see here is a translocation 517. So you can see that part of 5 has gone to 17, and the RARA, the 3' part of the RARA gene has gone and translocated with chromosome 5. Next slide, Ashish. Next slide. So, you know the PML RARA, but I will just go, and if you look on the right side, there is a whole list of variant translocation. So this one was an NPM1 RARA rearrangement, 5 and 17. So if you look, here is a whole list of different variant RARA translocations, and they're extremely rare. They are not so common. And sometimes we do joke that the number of number of variants defined are more than the number of patients ever seen with these kind of RARA rearrangements, right? 1 case, 2 case, and so on. Now, is it important to identify the partner? Next slide. Yes, it is, because you can see that some of the partners are resistant to your ATRA therapy. So, whereas PML RARA, FLIPI RARA, NPML RARA, and TFG RARA are sensitive, there are other patients like ZBT6 RARA or the PCOR RARA which are resistant to ATRA. And these patients then have to be treated like an acute myeloid leukemia. So, that is why it is important and essential to identify the partners. Yeah. Okay, can go to the next slide.
So, we can go to the.
Yes. So, any more questions? I can see there is a question. So, let me get a pen if you can see my video. Yeah. Okay, so I can see a question on the chat which says, kindly explain the break apart FISH. Okay. So see, generally, what is FISH? FISH is fluorescent in situ hybridization. So what happens there? Basically, we are using nucleotide sequences with a fluorescent tag attached to this. Now, these nucleotide sequences will go and bind to the complementary genes and give a signal. So usually what we do is, here what we have is the PML RARA. Let me start with that example. So the PML has been given, the complementary sequence has been given red, and RARA has been given a green fluorescent. So whenever they meet, you will get a fusion. Now what happens is, there are certain genes which have multiple partners. For example, your MLL gene or the KMT2A gene. Now MLL gene is very promiscuous. It has more than 100 different partners, more than 110 different translocation. So in these cases, how the probes are designed is, I don't know whether you can see my video. If you look at the pen and you have a cap of the pen, the probes are designed in such a way that the red and green are together where the gene is present. So just imagine the cap has the red and the green is present on the whole pen. And every time there is a translocation, the red and green will get separated. So these are called break apart probes, wherein you will see a yellow signal in a normal gene, and the red and green get separated whenever there is a translocation, irrespective of the partner. So the break apart probes are used whenever a gene is known to have translocation with multiple different partners, like the MLL I said. It has more than 100 different partners. So that is when a break apart probe is used for fishing. Any more questions?
So I have one question.
Yeah.
So, which is a better investigation, RT-PCR or FISH to detect the translocation? Like RT-PCR will detect the oncoprotein.
Yeah. So basically, what you must understand is in PML RARA, you need a quick diagnosis. RT-PCR would take at least a couple of days to come, which is quick. Okay. Now in RT-PCR, what you'll do is you're going to look for only 2 or 3 variants, the BCR2 and BCR3, and your primers will be designed based on where the break happens on the gene. Now, whereas FISH is much more broad-based, so you will be able to pick up a lot of variant translocations, right? So your RT-PCR will not be able to pick that up. So the first thing is when you suspect an APML, you need a quick diagnosis because we kind of treat it as an emergency. The patient can bleed anytime and the treatment has to be started as soon as possible. Blood protects are arranged for the patients, anticipating what if he has a bleeding episode, and the treatment has to be started quickly. So FISH, as I said, the result is given straight away within 4 hours. Now, in most of the patients, your FISH will be right and give you the diagnosis. Sometimes in certain patients where the PML RARA fusion is being formed because of an insertion of a gene rather than a translocation, your FISH will miss it. And that is where your RT-PCR will identify the product. Okay. Now, the question here is, suppose you've done your FISH is negative. So what we do is always extract the RNA of these patients, and then you do an RT-PCR and you can identify the PML RARA in an RT-PCR. Now, when you monitor these patients, an RT-PCR is a more sensitive technique, right? So it is always, so for diagnosis, straight away FISH it, and then you get your diagnosis. Suppose your FISH is negative, you do have an entity called as FISH negative PCR positive, wherein the PML RARA can be formed because of small insertions. And also in these patients, when you follow up, your RT-PCR is much more sensitive technique to follow these patients. We also use digital droplet PCR to follow up these patients, wherein you can look at the transcript, and the FISH can only look at 1 in 100 cells for positive, whereas your RT-PCR can go to 1 in 10,000 and 1 in 10 to the power of 6 is much more sensitive to follow these patients when they are being monitored during the treatment. Yeah.
Yes, thank you.
No more questions. We go to the next case, Ashish.
Yes, so we move on to the next case, case number 3. So this is a 61 years female present with a lower back pain since 1 year. And MRI of monocercal spine showed a degenerative disc disease, completion of L1 vertebra, and 2 small enhancing lesions in the iliac aspect of right sacroiliac joints, and a query of metastatic disease was given. At Tata, you had no lymphadenopathy, no organopathy, no color. Absolutely, the patient looked fine except for the low back. And on CTC, as you can see, there was no hemoglobin, the platelet was normal, the differential count was normal, and this is the peripheral smear. So, Dr. Rana, can you tell me what is the important finding in this peripheral smear?
In the peripheral blood, we see marked rural formation, and we see cells that look like lymphocytes. It has a vasofiltrum of cytoplasm and clumped chromatin. It looks like it's lymphoplasmacytic cells, or plasma cells, but look like lymphocytes. Yani multiple myeloma.
Okay, thank you, Dr. Rana. So, as you can see on peripheral smear, there is a mild ruling which is there, and you can see there are small. These are circulating cells, as we have already discussed that this kind of cells neither fall into the category of small B lymphoid cells or directly into the category of plasma cells. So, this looks like a lymphoplasmacytic cells. Dr. Sneha, are you there? If you can display what are the findings in the bone marrow?
So the bone marrow shows cells that have eccentric nucleus, moderate amount of cytoplasm, and they look like lymphoplasmacytic cells.
Okay.
There are a few smudge cells.
Yeah, so there are some smudging as you told that there are small lymphocytes kind of cells and there are lymphocytes which are eccentric nucleus and very small amount of cytoplasm look like lymphoplasmacytic cells. Okay. Again, you see there are cells which look like lymphoplasmacytic cells, or they are eccentric nucleus and plasma cell kind of cytoplasm, but did not exactly look like a plasma cell. And there are these small lymphocytes which are increased. On bone marrow biopsy, as you can see, there is complete effacement of the architecture, and you can see there are multiple aggregates of these small lymphoid cells. Okay, why do I call them small lymphoid cells? Because again, I've already told that these nucleus, as you can see, are very close to each other, so we know that there is a little amount of cytoplasm in them. And what happened to the ICs, as you can see, that CD20 highlighted these cells, which are small lymphoid cells. Again, CD130 was very strongly positive, CD3 was negative, and lambda was positive, and kappa was negative. So we know that these cells are CD130 positive, CD20 positive, and these are lambda restricted. So Dr. Asta, can you tell me, so now what will be your diagnosis based on this aspirate and biopsy?
The plasma cell dyscrasia, most likely multiple myeloma.
Okay, so that is it. So our first differential diagnosis will be multiple myeloma because 138 is very strongly positive. However, it is very unlikely to have multiple myeloma which look like a small lymphoid cells. If there be large lymphoid cells or blastoid looking like cells, so what are the other alternatives? So your plasmacytic lymphomas are also a good alternative because those will also show 38 positivity and some amount of 20 positivity. What are the further tests which were done in this patient? Again, serum protein electrophoresis showed an N spike of 2.1 gram per deciliter. The serum free light chain ratio, as you can see, is almost 0.001. 009, and you can see the immunophenotype electrophoresis again showed lambda restricted and immunoglobulin G restricted monoclonal pack. So we went on to do a plasma cell panel on flow cytometry. As you can see, this is a 11 color. This is a 12 color plasma cell panel or 11 color plasma cell panel. As you can see, there is lambda and kappa, which are cytoplasmic. So unlike the B cell malignancies or B cell lymphoproliferative disorders, in plasma cells you do kappa and lambda chain cytoplasmic staining rather than a surface staining, because in kappa the kappa and lambda light chains, they reside in the cytoplasm of the plasma cells, and there are certain gating markers. The CD138 and CD38 are used for the gating of the plasma cells, and there are certain antibodies which will be defining your plasma cells to be abnormal or normal, like CD56, which will be present almost in 70% cases of abnormal plasma cells. However, always remember there will be small subset of normal plasma cells which can also be CD56. So on the bone marrow biopsies, if you are doing only 138 and only 138 staining and 56 staining, you should also do a kappa and lambda staining because small amount of normal plasma cell can also express 56. And classically, the normal plasma cells will always express CD81 and CD27. However, your abnormal plasma cells will be negative for CD81 and CD27. Sometimes your plasma cells can also express in CD20 in 10 to 20% of the cases. As you can see, this is a flow cytometry experiment for a plasma cell neoplasm. Again, you get the singlets, rule out your doublets, do a forward scatter, side scatter, get only give the viral cells, and based on 45 band size scatter, just get your lymphocytes and granulocytes and monocytes, and your primary getting marker for plasma cells will be CD38 and CD138. So based on CD138, we know our plasma cells will be CD38 bright and CD138 bright. So you get your plasma cells, then refine your plasma cells based on 38 and 45. The plasma cells, abnormal plasma cells can have CD45 negativity, CD19 negativity. However, they will always be CD38 brighter. So you only take the population which is CD38 brighter population and CD45 dimmer to find out your abnormal plasma cells. As you can see, these abnormal plasma cells, which are given by CD38, 1 and 38, are negative for CD19 and 45. These plasma cells again are showing CD20. As you can see, there is a small cluster of these red cells which have CD20 positivity in the B cells. The plasma cells are almost all reaching to the level of normal B cells. So these are CD20 positive, 19 negative, 45 negative, and the cytoplasmic lambda restricted. As you can see, again, these plasma cells are CD117 positive. Remember that CD117, though is specific for abnormal plasma cells, they are only present in 10 to 20% of the cases. As I've already told, that CD81 will be present in normal T lymphocytes, B lymphocytes. However, they will be negative in the abnormal plasma cells. So your abnormal plasma cells are CD81 negative, and your CD27, as you can see, though these are abnormal plasma cells, they are also showing CD27. The classically, the abnormal plasma cells would be CD27 negative. However, CD27 positivity, if you get only CD27 positivity, it is not diagnostic of abnormal plasma cells. So you have to define abnormal plasma cells on the basis of multiple surface antigens like 19, 45, 56, 81, 57, 28, and as well as one clonality assay based on cytoplasmic kappa and lambda staining. So on the basis of this flow cytometry experiment, we know that my plasma cells are abnormal plasma cells. They are lambda restricted and also expressing 20, as we have already found in the bone marrow biopsy. So once we have done plasma cells, then we have to do a restratification for the patient based on certain molecular tests. So I'll request Mayur sir to tell you about how to do a restratification in case of multiple myeloma and how we did this in case of this case. Mayur sir, are you there?
Sorry, I was muted. So as you know, plasma cell, multiple myeloma is a plasma cell neoplasm. So plasma cells are mature cells. Now it is very difficult to get karyotypes out of mature cells. So because they're already mature, they're not spontaneously dividing. So one of the best ways to do a risk stratification for myeloma is to do FISH. So what we do is we take the marrow and we use CD138 antibodies, and we extract the CD138 positive cells using magnets, and then we perform FISH on slides made from this. So can you go to the next slide once, Ashish? Next slide. Next slide. Yeah, no, yeah, the genetics. No, behind. Yeah. So when you look at it, when you look at myeloma, so what may happen is there are something called as primary genetic abnormalities. So these genetic abnormalities happen at very early in the course of the disease, or the founding abnormalities. Now these founding abnormalities are trisomies or high hyperdiploids and immunoglobulin gene rearrangements. Now whenever a immunoglobulin gene rearranges, what it does is it has enhancer elements attached with it. So whenever it goes and rearranges with any other gene, it results in upregulation of that gene. So it can have different partners. So you have the 1416, you have the 1420, you have the 814, and so on. So based on the partner, what it can result is it can result in upregulation of the partner gene. Now these are the primary abnormalities. Now the secondary abnormalities are those that can happen any time during the course of the disease. So what will happen is as the disease progresses, you can have c-MYC abnormalities, you can have TP53 deletions, and you can have 1q gain and 1p loss, which are associated with a higher risk. So for every patient of myeloma, you risk stratify using cytogenetics. How do you do cytogenetics? You do cytogenetics by FISH. How do you do FISH? You do FISH by extracting CD138 plasma cells from the bone marrow, and then you do FISH, because as you know, multiple myeloma is a patchy disease. So if you just do a FISH directly on the marrow, you may do FISH, you may count your FISH on the normal cells because you just have the nuclei, and you may get a wrong result. So FISH on a myeloma has to be done on CD138 enriched plasma cells. Go back to the previous slide. Yeah. So what you see in this patient was that there was a 1114 translocation, which is CCMD gene was overexpressed, and this patient also had a 1q gain. Now 1q gain is associated with high risk, and 1114 is associated with a standard risk. Also 1114 is associated with a small cell myeloma morphology, and these patients benefit from venetoclax therapy, and that is why this genomic signature is essential to be identified. Next slide. So it's a small lymphocyte-like variant in myeloma. It is rare. It is seen in 3%. It's also called as a lymphoplasmacytic variant, characterized by CD20 expression and presence of 1114. As I said, it results in overexpression of cyclin D1. There is overexpression of B cell genes like CD20 and PAX5, and this is associated with a favorable prognosis. Next. So the risk stratification, the 4, 14, 14, 16, 14, 20 are the high-risk primary abnormalities. Deletion 17p, TP53 mutation, and 1q are associated with a higher risk. The trisomies, the 614 and 1114 are associated with standard risk. Whenever you have 2 high risk, it's called a double hit. And when you have 3 or more genetic abnormalities, it's called a triple hit myeloma. Next case. So you do have different morphological variants in myelomas. You have the plasmablastics, you have the flame cells, you can have the plasma cells having anaplastic features, you can have spindle cell features, and you can always have histiocytoid plasma cell features in a patient of multiple myeloma. Next. So any doubts on this case? Any questions? I don't see any questions, but if anybody has a question later, they can come back to us. We'll go to the next case, please.
So
I think Ashish will go a little quickly. It's almost an hour now, so that we finish the next 2 cases quickly. Yeah.
Okay, so this next case 4, 49 years male, present with pain abdomen, and CBC showed a very high TLC of 4.68 lakhs. The TLC clinically found a very used on ultrasound and on palpable, which is 6 cm below coastal line, mild hepatomegaly, and X-ray chest was into a respiratory distress, and bilateral pleural effusion was there. Now, CBC, we found out these kind of things. As you can see, there are increase in this blast-like looking like cells, I can say. There are cytoplasmic bleedings in this cell, as you can see. There are a lot of cytoplasmic bleedings, as well as a single punched out nucleoli in all of these cells. As you can see, there are single punched out nucleoli in all of these cells. So the TLC is very high, 4 lakhs, and you are finding a lot of these cells which are cytoplasmic bleeding. At some places, a nuclear irregularity, as well as punched out single nucleoli. On bone marrow aspirate 2, as you can find, these lymphoid cells are there at places which are well, again, punched out single nucleoli. On bone marrow biopsy, as you can see, this bone marrow biopsy is full of these kind of cells. And on IC, all of these cells are CD3 positive, 34 negative, TDT negative, 1A negative. So these 34 and TDT are immaturity markers. CD1A also helps as the immaturity marker in case of T lymphoid neoplasm. So you want to know if it is a T lymphoblastic lymphoma, which is of optical type, or which has arisen from thymic cortex. However, we have found that all these immaturity markers are negative and only CD3 was positive. So to find out if this disease is T lymphoblastic or is just a mature T cell neoplasm or T cell PD, we have to do some flow cytometry. As you can see, this is a dual color flow cytometer panel for T cell PD or T lymphoproliferative disorders. As you can see, there is a TCR alpha beta, T cell gamma delta. Sometimes we put CD30 also in case of T cell neoplasm. As you know that a lot of T cell PDs may have a CD30, which can be of diagnostic as well as of therapeutic significance. You have your CD3, you have 15 and 56 and 16 both in the same fluorochrome. If you put both in the same fluorochrome, you can use multiple colors simultaneously, and both 56 and 16 are positive in case of AK cells. We can get out all of my AK cells based on 56 and 16. As you can see, there is another marker which is TRBC1, which can be used to define clonality in case of TCR alpha beta T cells, and there are other markers which will be used for the current antigen expression such as 7 and 5. So in this case, again, we did a flow cytometry. The CD45, as you can see, almost all the cells are in the CD45 bright region. The lymphocytes are gated, and these cells are 7 positive. As you can see, these are brightly positive for 7. These are surface CD3 positive, and these cells are 2 and 5 also brightly positive. On 4 and 8, as you can see, there is a population which is co-expressing 4 and 8 or double positive for 4 and 8. There is negativity of 1. There is absolutely negativity for CD57, which is a large granular lymphocyte marker. These are negative for 16, again positive in large granular lymphocytes as well as in NK cells. And these are all of the cells are TCR alpha beta positive, TCR gamma delta hardly any cells. And these are again proven to be cytoplasmic CD3 positive, again proving that these are T cells, these are impure negative, TDT negative. So I know my Hematology markers are negative both on the bone marrow biopsies and IFCs and also on the flow cytometer. So 34 TDT and 1 is negative, which are Hematology markers. I have only the positive findings of that. This is CD3 positive, 7, 2, 5 are brightly positive, and these are co-expressing 4 and 8. So what about TCR C beta 1 or TRBC1, so which is a clonality marker? So as you can find, all of my cells are positive for TCR C beta 1 or otherwise called TRBC1. As you can see, there is single clonal peak of TRC beta 1. So how do we define clonality based on TCR C beta 1? As you can see, T cell clonality is classically done by TCR gene reaction studies or which is done by PCR based techniques, and there is a B beta reporter assay which is done by flow cytometer. However, both of these are very difficult and time taking. As we know, during maturation of the T cells, especially TCR alpha beta cells, there are two constant domains which are included in the T cells. Your T cells can either have a TRBC1 or TRB concentration 1, or it can otherwise have TRBC2. So your T cells will be divided into TRBC1 positive T cells or TRBC2 positive T cells. So if you run a normal peripheral blood and get all of your TCR alpha beta positive T cells, so it will both show TRBC1 population and another population to be TRBC2 positive cells. So if you only use TRBC1, you'll find one peak of TRBC1 positive cells and another peak which is negative for TRBC1. So as you can see, in case of normal cells like CD4 positive T cells which are alpha beta positive, you are finding two peaks. One is TRBC1 positive, one is TRBC1 negative. If you find out alpha beta positive and 8 positive T cells, again two peaks, TRBC1 positive and TRBC1 negative. So if you find both kind of peaks, then you know that your cells are polyclonal. However, if you are finding only TRBC1 negative or only TRBC1 positive cells, you know that your cells are clonal. So in our case, as we are finding only one TRBC1 peak, we know that our cells are clonal and they're expressing 4 and 8, and they have a morphology which is showing a punched out nucleolide. So 4 and 8 co-expression are very rare in case of T cell neoplasms. So you can either find them in T lymphoblastic lymphomas, which are of cortical type, where you will find 4 and 8 co-expression, but they almost always show a CD1A positivity, or you can find a 4-8 co-expression in 25% cases of TPLL or T lymphocytic leukemia, and very rarely in PTCL NOS or peripheral T cell lymphomas NOS. So, but to diagnose TPLL, we have some certain consensus criteria by TPLL International Study Group. What are the major criteria? Major criteria are there should be more than 5,000 cells of this TPLL phenotype in peripheral blood. There should be a T cell clonality by flow cytometry or by PCR. There should be abnormalities of chromosome 14 or chromosome X, or there should be higher expression of TCL1A or B or MTCP1. There are certain minor criteria. So you may have abnormalities in other chromosome like chromosome 11 or chromosome 8 or chromosome 5, 12, 13 as such, or there can be a complex karyotype. And there should be an involvement of TPLL specific site. Whatever TPLL specific site, you should have a splenomegaly and you should have a effusion. So either should have three major criteria or first two criteria, major criteria with one minor criteria. And we should remember there are again three morphologic variants. You have the classical morphologic variant where there will be a irregular nucleus, large cells with punched out nuclei. Sometimes it can mimic a small lymphoid neoplasm, or sometimes it can have a cerebriform nuclei, which you find in case of CJVC. 4-8 positive, again, I already said that can be found in 25% cases of TPLL. Complex karyotypes are common, and recurrent genetic abnormalities are there, especially inversion 14. And surrogate to inversion 14, you can also do a TCL1 expression by multi-parametric flow cytometry or flow cytometry, which is specific for TCL1A overexpression. So as we have more than 5,000 cells, and we have now known that this is T cell, the T cell clonality is positive based on TCR beta 1, we have to do a molecular study to find out if there are abnormalities in the chromosome which are specific for TPLL. So we will going to do a karyotype in this case. I'll request Marisa to now let you know what are the karyotype abnormalities.
So what you see here are two different metaphases from same patients, and this patient did have complex karyotype. And just like what is seen in a TPLL, we did have deletion 11q abnormalities, we did have an inversion 14, we did have an ISO 8q and 8q abnormalities were there. So it is kind of a classical case wherein all the chromosomal abnormalities associated with TPLL was present. So we were able to add additional information in confirming the diagnosis in this patient. Next slide, Carl.
Thank you, sir. So as Marisa already told that this karyotype shows all the classical abnormalities in which you find in case of a TPLL. So apart from the classical morphology, if you find this kind of karyotypes, especially when you have a complex karyotype and this kind of inversion 14 and abnormalities in chromosome 8 and abnormalities of chromosome 11, this is the classical case of TPLL and T lymphocytic leukemia. So if you have any question?
So Ashish, there is a question which says why there is a difference in the morphology of blasts in the peripheral blood and the marrow.
Okay, so our morphology on the peripheral smear and marrow will be different because on the peripheral smear we have a different staining, which is a right GM staining, and on bone marrow there is a different staining, which is a company provided hematology staining, which is again hematoxylin and eosin staining, but it is a company provided stain. So the staining is different, and the again staining by the peripheral smear is done automatically on a CBC analyzer in our setup, and all my bone marrows are stained manually. So the difference is based on the staining and the staining done by manual or automated method rather than difference in the cells as such.
Okay, I think there are no more questions. We move to the last case then, Ashish.
Yes. So we move on to the case 5. We'll do it quickly. So 42 years male, bilateral neck and axillary swelling with night sweats. There was a perrectal bleeding for 2 days, UAG abdomen, so multiple enlarged lymph node, efficacy of right neck swelling at outside diagnosed as non-Hodgkin lymphomas, and patient was referred to Tata. So at Tata Kolkata, we found that multiple cervical, inguinal, and axillary lymphadenopathy was there. However, there was no hepatosplenomegaly. On peripheral blood smear, we found that surprisingly the hemoglobin was raised. It was almost touching around 20 gram per deciliter. TAC was raised, and there was mild eosinophilia and mild monocytosis in the peripheral smear. So we moved on to do a further evaluation. So I know that RBC density has increased, and there was a suspicion of reactive erythrocytosis based on non-Hodgkin lymphoma was suspected. So first, the lymph node biopsy was done. As you can see, there was a complete replacement of the lymph node architecture here, which is almost completely effaced by these blastoid-looking like cells which are there. And these cells all are positive for 7, very strongly positive for 7. They are very strongly positive for against CD3. They are negative for CD20. Ki67 was very high. CD34 was negative. Again, you can see almost 30 to 40% of the cells showed a TDT positivity. 4 was strongly positive, as well as 8 was also positive. So a fair number of cells out of these also showed CD8 positive. So yes, there was a co-expressing CD4 and 8 positive population. And again, CD1A was positive. As I've already told in the previous case that whenever CD1A is positive, CD1A can be taken as a immaturity marker in case of T cells. So as CD1A is positive, 4 co-expressing cells are there, and TDT is positive. So we know that we are dealing with a T lymphoblastic lymphoma, which is of cortical type. So once you have diagnosed T lymphoblastic lymphoma, these T lymphoblastic lymphomas and B lymphoblastic lymphomas, they tend to involve your marrow also. So in the marrow involved, so we have to do a marrow now. On peripheral smear, we did not find any blast. And on marrow, as you can see, the marrow is very hypercellular. You can see the fragments, it is almost completely packed with cells. So this is a hypercellular marrow, which shows a lot of myeloid cells as well as megakaryocytes. In the bone marrow biopsy, you can see there are a lot of megakaryocytes as well as myeloid cells. As you can see in the lower down in the picture, the arrow cells are all eosinophils. So there are a lot of eosinophils which are there. So there is a bone marrow eosinophilia. We had a peripheral blood mild eosinophilia and mild monocytosis. And then surprisingly, when we did CD34, it was negative, and CD117, which was highlighting these cells. So there are certain cells which are spindle appearance and highlighted by CD117, and 41 highlights the megakaryocytes. So Dr. Asta, can you tell me what are the cells which are highlighted by CD117 here? Okay. So you know the CD117 highlights the cells which are there in the marrow. What are the cells which are highlighted by CD117 in the marrow? So your pro-erythroblast can be CD117 positive, which is usually deemed positive for CD117 in the bone marrow. You can have your pro-myelocyte, which can be deemed positive for CD117 in the bone marrow. You can have your blast, which can be positive for CD117 in the bone marrow. And again, there are mast cells, which are always strongly positive for CD117. So mast cells are the brightest positive for CD117, both in ICs and in flow cytometry. As you can see, these cells are very strongly positive, has a spindle appearance. We know these are mast cell clusters. So these are mast cell clusters which have a atypical morphology. So you have a marrow which is hypercellular, showing bone marrow eosinophilia and showing this mast cell proliferation. So does this finding ring a bell in you? Yes. There are certain neoplasms which can have T lymphoblastic lymphoma, simultaneously have a myeloproliferation. So the myeloproliferation, we had a polycythemia, along with that a bone marrow eosinophilia, and there is atypical mast cell proliferation. So what are the neoplasms? These are in 2016 WHO called as myeloid and lymphoid neoplasms with eosinophilia and rearrangement of PDGFR alpha, PDGFR beta, or HGFR1, or with PCM1 chat2. The classification has been changed and renamed in WHO 2022, which is now called the myeloid and lymphoid neoplasms with eosinophilia and tyrosine kinase gene fusions. There are certain novel variants which are now included, such as FLT3 arrangement or ETB6-ABL1 fusion or the defined tyrosine kinase fusions. So I'll request Mayur to tell you about the karyotypes which you will find in these cases.
So what we found in this patient was a translocation 813. So your FGFR gene is present on the short arm of chromosome 8. So now here we had a patient which showed T-cell lymphoblastic lymphoma along with eosinophilia and a myeloproliferative neoplasm. So when we did karyotype, we found an 813, and since the FGFR1 gene is present on short arm of chromosome 8, we knew we were dealing with a next slide, Ashish. Next slide. So we are dealing with an 8. Next slide. So we're dealing with what is called as an 8P myeloproliferative syndrome, or an 8P syndrome, which is also called as a stem cell leukemia on lymphoma. It is rare, it is aggressive, and there are 17 different partners. This can be with or without eosinophilia and monocytosis, and they do present with TLBL. These are very high risk cases. These patients don't do well, require a bone marrow transplant. FGFR inhibitors can be used for these patients. Midostaurin can be used, and a lot of novel FGFR inhibitors are now available. But in spite of that, the outcomes in these patients are not good. So I think before I thank everybody, this was the last case. There's a question for you, Ashish. Why CD41 was done? It normally stains megakaryocytes. What is the need in this context?
Okay. So now we've known that there is a bone marrow eosinophilia, probably along with the polycythemia, bone marrow eosinophilia, and increase in mast cells, probably we are dealing with a myeloproliferative neoplasm. So the most characteristic finding in case of myeloproliferative neoplasm or the classical MPS are based on the megakaryocytes rather than other cells. So if you remember your polycythemia, your essential thrombocythemia, or myelofibrosis, the characteristic feature is distinguished based on the megakaryocytic morphology. But in this case, we did not find any megakaryocyte cluster. To find out if there are any abnormal megakaryocytes which are there, which clustering could not be seen on the naked eye, so we did a CD41 stain just to highlight the megakaryocyte if there is any abnormalities in the megakaryocytes.
Yeah. So, sometimes on morphology, you can miss the micromegs. And as Ashish was saying, the CD41 would just highlight the megs. And you can see focal clustering and help you with additional evidence for diagnosing your case of myeloproliferative neoplasm. I think if there are no more questions, I would like to thank Ashish. Ashish, first, most of all the slides have been made by him. I've just been here talking. And I would like to thank all the postgraduate students who joined us, Dr. Rana, and Dr. Neetu, Dr. Himani, Dr. Asta, Dr. Sushma, all of you. Thank you for joining us. I would like to also thank Dr. Gujral and Dr. Bojes. Gives us an opportunity to interact with so many students on a single platform and share our work and experience. If there are any more doubts, we are still here. And I think, Ashish, you can show the last slide of our lab to the people. Any other comments or questions?
So I think no other questions are there for today.
So, okay, so this is our hospital town, what you see on the picture. And this is, we come on traditional attire on one of the festivals, the Durga Puja, which is the biggest festival in this part of the country. And yes, that's Ashish, that's our lab director, Dr. Mishra, and this bright young gentleman there is me. Yeah. Okay. So thank you, everybody, for your attention. Thank you. I think we'll close the session then.
Yes, sir. Thank you, Dr. Mayur
Thank you,
Parihar. And
Dr.
yes, sir. Thank you, sir. Thank you for the elaborate case discussion on lymphomas and leukemias. Thank you, sir. Thank you for your wonderful talk.